Hello everyone, my name is Ian McLaughlin, and I have a PhD in neuroscience from the University of Pennsylvania, where I focused on mapping out some of the neurobiology of addiction and anxiety, and I'm really a neuropharmacologist at heart, right? Which means that I really approached neuroscience because I was interested in how various molecules influence consciousness by interacting with the brain. And one of the topics that's been consistently intriguing to me over the past several years has been the pursuit of novel pharmacological strategies, meaning like novel medications, to treat psychiatric conditions. Specifically, one of the more intriguing efforts, at least from my perspective, has been the evaluation of a substance called psilocybin for the treatment of PTSD. Okay, so let's start with a little bit of background. So first of all, the topic for today implicates an entire family of substances, right? The psychedelics, not just one. And so this is a family of compounds that, share similar psychoactive effects and, with some variability between them, of course, right? They're different molecules. But given that similarity in their effects, they unsurprisingly also share some of the same pharmacological action. And by pharmacological action, what I mean is what it is that these molecules do when they get into the body, and then, more interestingly, what they do when they get into the brain. So there are a variety of ways that any given molecule can interact with the brain to produce psychoactive effects, right? The simplest, probably, is when a molecule is capable of binding a receptor in the brain that's typically bound by one of the neurotransmitters that the brain produces or receives via, your diet, right? So and neurotransmitters are basically just molecules that the cells in the brain, neurons and glia, release, right? And it's one of the ways that messages are sent between the cells of the brain, right? And so, if you're watching this, you're probably familiar with a few of them: dopamine, norepinephrine, or noradrenaline, oxytocin, and then, most importantly to our conversation today, serotonin. So I'm not going to turn this into a whole intro into neurotransmitter signaling, because believe me, that's a gigantic topic, and it's going to be more complicated than I'm going to make it sound. But just keep in mind that essentially every neurotransmitter that you can name does many things, right? Essentially no neurotransmitter does just one thing. And so there's two reasons that I want to emphasize that fact, beyond just I think it's one of the more common misconceptions about how neurotransmitters work. But the fact that, essentially no neurotransmitter does only one thing is important because, you often see neurotransmitters described as the love neurotransmitter, particularly when it comes to oxytocin, or the happiness neurotransmitter, when it comes to dopamine, as well as serotonin sometimes, which is sort of a hint that happiness is more complicated than just the levels of one neurotransmitter, right? It's just not how it works. It's not how consciousness works. And so basically every neurotransmitter is involved in multiple aspects of our conscious experience, and can be involved in aspects of our consciousness that would intuitively seem maybe completely unrelated. So for example, dopamine, right, is indeed involved in happiness. As hard of a thing as that is to even define, what do we even mean by happiness? But it's really centrally involved in the expectation of reward and salience. So don't worry about that. But it's also involved in regulating appetite. Right. And so, there's a very good argument that a competent, neuroscientist could make that reward and appetite are intrinsically associated with one another. Right. Given that access to food is one of the most fundamental dynamics that governs survival. Right. And therefore evolution. Right. But, I suspect that at least a bit of familiarity with neuroanatomy would be required for someone to actually, competently trace the pathways that, between dopamine levels in a certain brain region like the ventral tegmental area or the nucleus accumbens, trace that activity to fluctuations in appetite, which are regulated in different parts of the brain. So, don't worry about all these names and this terminology. The point is that, there are different aspects of behavior and conscious experience that can be influenced by a single neurotransmitter. And so the same is true for serotonin, right? And perhaps even more so. Serotonin signaling regulates a huge variety of aspects of consciousness, from, mood to sleep to cognition, in the way we think, right? To appetite, and then relevant to the conversation today, sensory perception, right? So let's take a look at a molecule that might be considered a classic psychoactive or psychedelic substance, psilocybin, right? That's psilocybin right there, right? And you don't really need to have any familiarity or experience in organic chemistry to see that these two molecules, right, psilocybin and serotonin, they're pretty similar shapes, right? there's some differences, obviously, but, those differences, and those differences are important, right? But they're clearly like similar shapes, right? You have like a hexagon connected to a pentagon, and then you have like a squiggly line that connects to an N that's connected to H, right? And then, serotonin has this OH right here, a hydroxy, and psilocybin has this P surrounded by a bunch of O's, right? A phosphoryl group. And so, those are some differences. But for sure, quite a few similarities when it comes to, imagine all of the ways that a molecule can be arranged, right? And as a result of this similarity, this molecular similarity with serotonin, psilocybin, which, by the way, is found in a variety of, different types of mushrooms, perhaps most commonly the Psilocybe cubensis mushroom, which is right there. But it's due to that similarity with serotonin that psilocybin can sort of like masquerade as serotonin, once it gets into the brain. And it's not technically psilocybin once it gets into the brain, but don't worry about that. It can do a lot of the same things, right, that serotonin does. It binds many of the same receptors that serotonin binds, and it can influence how serotonin itself, the molecule itself, moves around throughout the brain, right? And because serotonin influences so many aspects of our conscious experience, psilocybin also influences many aspects of our conscious experience, right? And, probably the most well-known effects of this substance and the whole family of substances are their effects on sensory perception, right? And particularly visual perception, right? They're technically hallucinations, but that term is a little bit inadequate when we're trying to describe the kind of quality of the effects of these substances, because, there are different types of hallucinations. So like generally speaking, like all psychoactive substances, this will vary between different people who have different brains, obviously, but, generally speaking, substances like psilocybin don't induce like 100% completely fabricated and super realistic perceptions, hallucinations. Generally speaking, the types of hallucinations that people experience on these substances are more like, you can think of them as like modifications of existing sensory perceptions, right? You're not like interacting with people who aren't there, right? In a way that's like indistinguishable from your, real life, your normal life, your sober life. It's not like you'd see someone sitting next to you, right? Just like you would when you're sober, who isn't there, right? Those kinds of hallucinations are associated with a different class of psychoactive substances called deliriants, okay? The hallucinations of deliriants are like uniquely realistic. And so when it comes to the psychedelics, the sensory distortions aren't particularly realistic. These substances change the way that incoming visual, stimulation is perceived, is understood, right? And, there's a connection to reality that's sustained under the influence of these substances, but reality is definitely very modified, right? And of course, we're talking about the typical doses that people take, right? We're not, to achieve these effects. We're not talking about like the gigantic doses that are sometimes consumed. And by the way, I talked about a fairly recent story of someone who accidentally snorted 550 times the normal dose of LSD on this channel in the past, and I put a link into the description below if you want to hear about like what super high doses of these substances can induce. And the person turned out to be okay, interestingly, but if not a little bit jarred. But anyway, so some examples of the effects that people can experience on a psychedelic. Color perception is modified, right? Walls can begin to sort of oscillate, can begin to breathe. There can be this perception of geometric shapes and patterns that, it's stuff like that aren't actually present, when looking at something like a popcorn ceiling, those textured ceilings or like a textured wall, there's like little subtle kind of topography to the wall, right, that you don't even really notice when you're just looking at it. But under the influence of these substances, there can be this effect where you begin to see like geometric shapes emerging. And then, things in the visual field can sort of drift, to and fro a little bit and wiggle, right? Distortions of things that are actually there. And so, interestingly, like, the same kind of thing happens to other sensory perceptions beyond just visual, right? A person's auditory perception, for example, like, what they're hearing, can be modified. And by the way, and I'm totally speculating here, but I suspect that this family of substances, psychedelics, can actually induce more realistic auditory hallucinations relative to what they do in the visual system, but that's a topic for another day. Anyway, so, the kind of sensory, of distortion of sensory perception, right, can also influence things like your sense of touch. If someone, for example, looks at a cactus, right, or something with, a real serious texture, they can sort of feel the prickliness. And they can, these substances can even influence, subtler aspects of our conscious experience, right? Time perception is very regularly completely distorted, right? So like a minute can feel like an hour, or 5 hours can feel like 10 minutes, right? Just warped time perception. And, some of the more interesting effects, at least from my perspective, someone who's like a card-carrying nerd when it comes to being interested in understanding consciousness, the more interesting effects for me are the effects on cognition and on mood. Okay, so these substances alter the way that cognition operates. Okay, thoughts and ideas can bubble up that had never happened, when not under the influence of these substances. And emotional associations with, a huge variety of things is altered, right? People can like have overtly emotional experiences from like looking at an inanimate object, right? Inanimate objects can become like weirdly personified or anthropomorphized. They can take on character, right? So strange cognitive associations can happen. Like you can have like an association between a memory that you had about a trip you took with your parents when you were 11, when you're listening to some new song that your favorite artist just put out that you've never heard before, right? Just sort of weird connections. And when it comes to mood, the substances can influence, can induce all kinds of fluctuations in, your emotional experience, right? Fluctuations between like elation to sadness to euphoria to, empathy and contentedness, on and on, right? These like kind of warped, huge fluctuations in mood, right? The substances can be emotionally pretty volatile. And I can go on and on, right? The effects of these substances are varied and very complex. But the fundamental point I'm making is that these substances influence a wide variety of aspects of our consciousness that extend well beyond just the visual hallucinations for which they're most well known. And it's part of that, variety of effects that has inspired or like, maybe revivified, right, an interest in the potential psychiatric applications of substances like psilocybin, but then the whole family of psychedelics. And so there's a super interesting backstory, right, to the attention that this family of compounds has received, right, from folks like Tim Leary, who was a psychologist who was interested in, the potential clinical utility of LSD and psilocybin, and a whole bunch more. He went way beyond just that introductory interest. But then other folks, right, like Sidney Gottlieb, whom you've probably never heard of, but he was the director of the Division of Technical Services Staff at the CIA, Central Intelligence Agency here in the United States, and he was involved in essentially running projects like MK Ultra, and among other wild and like deeply unethical endeavors by the CIA in the 50s and the 60s. And if you're interested, like if you'd like to dive into that whole history, right? What I think one of the best books that's covered this, like the whole involvement of the CIA with psychedelic compounds. I think the best book is this one called Poisoner in Chief by Stephen Kinzer. And so Stephen Kinzer is like a highly accomplished journalist who's been a professor at Northwestern University, Boston University, and he's now a senior fellow at Brown University, right? Well-regarded institutions. And it's a super fun book, right? Particularly, if for anybody who's interested in history in general, but particularly the history of like the United States and or the intelligence community, and then in that era when the CIA was like interested in developing mind control, quite literally. Okay, so anyways, I'll suspend the nerdery there, but my point is that these substances have a pretty long history in the United States. Most recently, however, after this long period of being relegated to being as illegal as a substance can possibly be in this country, this family of substances is enjoying renewed interest from biomedical science. And so specifically, there's a study that's being done by an organization I brought up now, several times when talking about these substances called MAPS, right? And this study is being conducted at Johns Hopkins University, right, which is one of the best biomedical institutions in the world. Also, by the way, when it comes to Johns Hopkins, it's probably singly responsible for making medicine in the United States an actually like respectable discipline, an effective discipline, rather than being laughable relative to medicine in Europe basically at the beginning of the 1900s. There's another tangent that we'll avoid, but it's an interesting history as well. Anyway, so MAPS is conducting this study at Johns Hopkins, right, to explore whether psilocybin can produce, meaningful, deep, emotional, important, right, experiences in cancer patients. And this study that's ongoing is a follow-up to a study that had been done in the past, which was a randomized waiting list controlled clinical trial that was also previously done at Hopkins. And that study found that the substance was able to produce large, rapid, and sustained antidepressant effects in patients with major depressive disorder. Okay, so, this isn't just happening in the United States. There are other trials that are being conducted in the US, but also in Europe, that explore whether psilocybin might be beneficial for the treatment of alcohol or tobacco addiction. And then in a completely or mostly unrelated domain of medicine, psilocybin has been found to be rather effective for the treatment of various headache-associated maladies, right? Migraine and cluster headaches in particular. And there's this whole body of research that explores, the effects that this family of substances has on neuroplasticity, and a little bit more on that later. But the point is that there's this track record of research that suggests that these substances may have some psychiatric utility. So that brings us to the study that I'd like to discuss today. So this is actually a pretty complicated study, okay? And it strikes me as being more of like a technical proof of principle, right? They like invented a new technology basically, but it's definitely relevant to this effort to find therapeutically relevant substances that might be useful in the treatment of psychiatric conditions for which we have pretty much like few effective pharmacological options right now, right? And what they did is definitely complicated, and it is impressive, but just stick with me. It's totally understandable. Okay, so this group developed what they call Cyclite. Okay, and really, this is what this technology is. Okay, it's an engineered neurotransmitter receptor. Think a serotonin receptor that serotonin itself might bind that you have in your brain right now. Or it could be a dopamine receptor or any other kind of receptor, right? But they modified it so, in a way that basically lets scientists know if it's being exposed to certain substances that are capable of binding the receptor, right? So it's sort of like if you had a light in your living room, right, that only turns on when a specific person is at the door, right? I can imagine having a light on my desk that only turns on if my baby is moving a lot in her bed, right, while she's, supposed to be sleeping, so the light is never on unless she's in bed. And, then she's also moving around a lot. That's the only time that the light is on. That's how I know that she's moving, right? That's sort of the basis of this technique. And then beyond that, like more usefully, imagine this light again, right? If she's only moving a little bit, the light is kind of dim, right? It's on, but it's dimly lit. If she's tossing and turning a lot, around a lot in bed, likely about to get up and maybe wander out of her bedroom and like freak me out at my desk in the middle of the night, the light is super bright. Okay, that's kind of what we're talking about, right? With Cyclite, if a molecule just sort of kind of interacts with the receptor, not very intensely, doesn't really bind it that strongly, there's a dim signal that scientists can detect. But if a molecule gets like a hell of a grasp, it really binds really hard that receptor. It's like you're standing under fluorescent tubes in a grocery store or something. It's super duper strong signal, very bright, and so. This is actually based on an older technology. It's like an iteration on an older technology. But the point is that scientists can detect whenever, a specific serotonin receptor in the brain interacts with a molecule. And the receptor that the scientists have honed in on is the one that we think that most neuroscientists believe and have good reason to believe is responsible for like the vast majority of those sort of wild hallucinogenic effects of the family of psychedelic compounds that includes everything from psilocybin and dimethyltryptamine or DMT to mescaline and LSD, right? It's the 5-HT2A receptor, and so that's the kind of serotonin receptor it is, the subtype. So the group did a whole bunch of fun science and using techniques actually that the lab in which I was working was just beginning to establish by the time I finished my PhD. Really useful stuff like vibrophotometry. But the group also seems to have produced a rather sensitive sensor. Okay, and that's really what this paper is about. So they produce this technique that will enable scientists to understand when a molecule interacts with a very specific serotonin receptor subtype. And that serotonin receptor happens to be the one that we think is responsible for the majority of the hallucinogenic effects of substances like psilocybin and LSD. So the innovation is really about this new technology, right? Not really about any one particular clinical application, but there are potential clinical applications, right? And when I say applications, I mean like drug development, like the development of new medications. And so I referenced, findings that indicate that these substances have already, as they exist today, have some clinical utility. And I discussed some of those findings in the past on this channel. But a pretty central limitation to the psychiatric use of these substances is the fact that they induce hallucinations, psychedelic hallucinations. And, some people clearly, very clearly, really enjoy that effect, right? They, use them recreationally. To many people, that's like a really unpleasant and maybe like prohibitively aversive side effect, right? And I don't blame, I wouldn't blame somebody for being kind of weirded out by the prospect of experiencing all kinds of hallucinations and stuff, particularly if they're confronting something like terminal cancer or depression or PTSD or, any kind of psychiatric condition, right? They have enough on their plates. They really want to like sign up for having that plate, that they have that's already full, take on this whole, new personality, right? Or have their ego dissolve or have the walls breathe, right? That's pretty jarring. So what this paper is really about is that, this technique that they created that'll enable, scientists to understand when a molecule interacts with this serotonin receptor subtype. It's a very useful innovation and not a simple one, but I'd be willing to bet that they did not necessarily set out to discover a molecule that's capable of producing the effects that they ended up finding, right? That's capable of delivering a lot of the same effects of a substance like psilocybin, but free from the hallucinogenic effects. So beyond just the technology, they did find something that was rather remarkable. A molecule that does seem to deliver many of the same effects of, again, substances like DMT or LSD, or even structurally different molecules that don't look very much like serotonin, like DOI, which is sort of like a really super duper psychedelic version of amphetamine. You can kind of think of it that way, but without the hallucinogenic effects. And so, a substance that might be useful in a psychiatric setting, for example, right? But, doesn't induce hallucinations that, many people would find intimidating at the least, or maybe just completely disqualifying, like, no, thank you. And so, to be honest, the reason that this paper was published in a journal like Cell, this paper, which is, to be clear, is an extremely prestigious scientific journal. The reason it was published there is almost certainly, at least in part, due to the fact that they're exploring this receptor that's, thought to be responsible for hallucinogenic effects of many psychedelic substances, right? It's sort of a buzzwordy kind of topic to be exploring. But by using this tool that they, innovated, they seem to have identified this molecule that can, again, deliver many of the beneficial effects that psychedelics can induce without the hallucinations. And so they had this sensor, right, that could detect or could signal to them whether a drug interacts with this specific serotonin receptor. And so they tested a library of molecules, 34 molecules, right, to be specific. And, just to see if, how these molecules would interact with the 5-HT2A receptor, right? That serotonin receptor subtype that we think is responsible for the majority of hallucinations. And so they found a specific molecule, they call it AAZ, A154, and they found that this molecule induced what appear to be potential antidepressant effects, as well as psychoplastogenic effects, meaning basically just effects that induce neuroplasticity. And just to be clear, that title, AAZ-A154, that is not an unusual kind of inscrutable, a designation for a novel molecule before it has any kind of known characteristics, right? Oftentimes, people will go by these kinds of just like letters and numbers. And so here is one example of how they demonstrated this psychoplastogenic effect. So here's what you're looking at, okay? And again, we're talking about essentially neuroplasticity. So in one of their figures, this is just part of a bigger figure, they have a picture of two neurons. That's what you're seeing here in blue. And one of the neurons was treated with some kind of like placebo, saline probably, or PBS or something, basically salt water, that's kind of like the fluid in which neurons typically live, right, in the brain. And then the other neuron up here, VEH, vehicle, was exposed. Or I'm sorry, the other one is AAZ. The other neuron was exposed to AAZ. Sorry, I'm hearing something kind of weird. So you don't have to know, you don't have to like understand a lot about neurobiology to see that this neuron, the one on the bottom, right, the blue thing is a neuronal cell body, the center of the neuron, but this one on the bottom, which was treated with their novel molecule AAZ, has quite a few more of those little tendrils, right, that are stretching out from that central circular cell body. Those are dendrites, okay? AAZ seems to induce greater dendritic branching, which basically just means it would be able to connect with, or form more connections with neurons more readily than the one that was just treated with vehicle, right? So it's inducing or amplifying neuroplasticity. And so they did some other experiments that basically demonstrate that the substance, AAZ, seems to do this via activation of that very serotonin receptor, right? Because when they prevented it, the molecule AAZ, from interacting with that specific serotonin receptor, they no longer saw that increase in the number of those tendrils. So super cool. So why might you want a substance that amplifies neuroplasticity, right? Well, when someone's engaging, psychological counseling, it might be the case that, the neurological consequences of the therapeutic effects from just counseling, from just psychological counseling, they may take effect more rapidly, right? If the connectivity between neurons, if neuroplasticity is primed, to change more rapidly than, this connectivity otherwise would. And so, sorry, I'm hearing what sounds like an electrical short, which is freaking me out. Okay. Anyway, sorry about that. So they back to neuroplasticity. They compared some of the effects of on neuroplasticity of their substance AAZ to a molecule called tabernanthalog. So tabernanthalog is an analog of a molecule called ibogaine, which comes from a tree native to Africa called Tabernanthe iboga. Okay, so tabernanthalog. And I've discussed ibogaine, in the past as well on this channel, and which also seems to present some potentially like dramatic psychiatric benefits, including remarkable effects that could potentially combat addiction. But it also, much like psilocybin, but even worse, can suffers from the fact that it induces an added effect of about a 35 to 48 hour long hallucinogenic effect, right? So that's a long time. You're going to sleep hallucinating, you're waking up hallucinating. That's pretty intense. And so tabernanthalog is sort of like a chemical iteration on ibogaine, right? And that's, similar to the effort in this study. But their evidence suggests that this molecule that they identified, AAZ, may be even more effective in some of the effects that are presumed, to be, psychiatric benefits, right? Some of the effects that are presumed to be, beneficial in a psychiatric setting. And so another really cool aspect of this paper is that, the molecules can be tested really easily with this technology. Don't need to work with any animals, again, think of the light that I wish I had on my desk that would tell me how much my baby daughter is moving in bed, right? If it's not on, she's not moving. If it's dim, she's moving a little bit. If it's bright, and she's up and about to surprise me at my desk, right? And so this tool could be used to get a sense of like how much any given molecule interacts with, the receptor, again, that we think is responsible for the majority of hallucinogenic effects of psychedelics. And so, beyond even that, the team may have identified a molecule that fits the bill, right, of a substance that does deliver those effects, but without the hallucinogenic effects that aren't exactly appetizing to everybody. So all of that said, all of our admiration stipulated, right, this is a super preliminary paper, to be clear. This is the kind of research that I did in the lab, right? They're working with animal models, not with humans, and they're sort of making some assumptions, and maybe the most important assumption that they're making is that it's the activation of a very specific serotonin receptor that's responsible for the hallucinogenic effects of this family of substances, right? Again, that 5-HT2A serotonin receptor subtype. And so, to be clear, like I would absolutely make the same assumption, right? But it's just difficult to ask a mouse or a rat or any other animal if they're like experiencing hallucinogenic effects, right? If the walls are breathing. You can't just ask them that. And so they evaluated whether, the animals were exhibiting sort of the typical behavioral signs that they tend to exhibit when they're exposed to things like, psilocybin or LSD, right? These are unique behaviors that they really generally only exhibit when under the influence of certain substances like this family of substances. But, it's just not the same as asking a human if they're seeing the walls breathe, right? And so, there's a ton of critiques that I could go through, that I would make if like they were in my lab, or if I were working in their lab even, about specifically like how widely their virus was expressed in many of their images. And so they use a virus to drive, to induce the expression, to basically implant this chemical, let's call it this, Cyclite, their technology. Some of these images looks like they were just like filling up the brain with their virus, which would not have flown in our lab. But, the reality is I do think that they're ultimately correct in their interpretations of their results, just given the data that we have at our disposal, right? At this stage of research. And so, by the way, I think it's just worth acknowledging that, this work was funded by something called the Brain Initiative, which is this whole effort through the National Institute of Health in the United States the Brain Initiative, the goal is to amplify the development of like novel technologies to interrogate the brain, to explore the brain, to understand consciousness, and develop new therapeutics, new medications. So anyways, the takeaways here. The group, developed this technique that enables scientists to get a sense of when any given molecule interacts with a specific serotonin receptor subtype, the specific serotonin receptor subtype that we think is responsible for the majority of the hallucinogenic effects of some psychedelic molecules, the same psychedelic molecules that appear to be, as of today, right, quite effective in treating certain psychiatric conditions, and probably, I think it's safe to assume that they'll probably be useful for treating more than just the conditions that have been tested so far, right? PTSD, depression. But beyond that, the team does appear to have identified a molecule that may, deliver many of the same effects that these, psychedelic substances induce. The clinical effects, the psychiatric, the effects that are useful in a psychiatric setting. But it doesn't appear to induce the same hallucinogenic effects, right, that those substances do. And so it is a bit of a holy grail kind of a finding in terms of neuropharmacology. And even if this particular molecule that they honed in on doesn't end up being useful for psychiatric applications, this tool that they made, Cyclite, may help to discover additional molecules, right, in the future that could lead to completely novel pharmacological strategies to treat some of the most difficult conditions, right, that millions of people confront, conditions for which we have only like marginally effective treatments, and those treatments that we have today have a wide variety of side effects that have a very real effect on quality of life. And so it's an exciting, innovation, particularly if you're interested in this sort of family of potential, psychiatric substances. That's what I have for you today. If you dig this kind of a discussion, maybe you'll consider subscribing. Otherwise, I will see you next week. Is that why SSRIs and SNRIs affect appetite? Yes, Scott, absolutely. Good to see you, man. And yes, and thank you, Cosmic Rose. I appreciate the likes. But yeah, that's why, I'm sure you're talking about the fact that like serotonin, influences more than just mood, right? If that's all that serotonin influenced, then we would have cured depression, right? Because SSRIs like, Prozac, fluoxetine, there's a huge list now of SSRIs. They do very effectively raise serotonin concentrations the first time you take them, right? It's not like MDMA or a substance that like really cranks up serotonin levels radically, but, like I've measured it in the lab myself. Like you can see it's just this steady increase, sustained elevation of serotonin that lasts for a pretty long time. And so if depression, were just a byproduct of insufficient serotonin levels, which is what used to be the sort of prevailing, theory, then we would have cured it already, right? And so, clearly depression is more complicated than just serotonin, and that's now widely appreciated at least in neuroscience. But also serotonin does more than just regulate mood, right? Given the fact that these medications that really only elevate serotonin levels, some of the other monoamines as well, they have a bunch of other side effects as well, right? They can influence sort of energy, cognition, libido, a huge variety of things. And so I should note, like, these substances they're kind of known for inducing weight gain, right? And that is in part due to, their effects on appetite, but it's not exclusively, right? So, and this isn't only antidepressants, particularly atypical antipsychotics can induce pretty substantial weight gain. And it is, again, in part due to how they regulate appetite, but it's even a broader effect on weight and metabolism. They alter the way you mobilize caloric energy basically in the body. And so you end up just packing on more weight. And so it can be, not only is it more difficult to lose weight, right? And this isn't true for everybody. This is true for some folks. They experience this side effect. But so not only is it more difficult to lose weight or keep weight off because, your appetite is affected, but it's also quite literally more difficult to lose weight, even if you're doing, an hour of cardio every day, right? It's not a huge effect, but yeah, that is a significant, factor in somebody's quality of life. Good instincts there. All right, I will see you all next week. See you guys.